Okay, this podcast is going to go over central dogma. Central dogma is a fancy word for the process of how you go from DNA to RNA to protein. So this whole process of going from one molecule to another and then to protein is called the central dogma. So here on this first slide is just an overview of this process. You have our double-stranded DNA. Then we have our single-stranded RNA, which is smaller. This process right here of going from DNA to RNA is called transcription. Transcription comes from the word transcribe, which means to copy. It's like saying I have a cookbook. The cookbook is too big, so I need to transcribe it onto an index card. That is essentially what is happening here. Then the RNA is going to be used along with ribosomes to create a chain of amino acids, which is your protein. So when these amino acids are folded, they form protein. So that's the goal, going from DNA to RNA and from RNA to protein. So again, I'm just going to repeat myself. Transcription comes from the word transcribe, which means to copy. Okay, you're copying something, just like you copy notes from a textbook. It's called transcription. That is the first part of DNA of the central dogma. Okay. Transcription, again, why do we even need to do this? Why do we need to go from DNA to RNA? And the reason why is because DNA cannot leave the nucleus. Okay, so here it says help, I can't get out. So what you do is you make a messenger, and that messenger is going to be RNA. We call it mRNA just because it's going to leave and it carries a message out with it. We see the ribosome there, so it's going to be very important in the next part. So before we go to this next part, I want to go over step 1. In order to make RNA, the DNA, as you see here, this is TATACG. This is DNA. The DNA is going to unwind just like it did during DNA replication. And RNA polymerase is going to come in. This is not DNA polymerase. This is RNA polymerase, and it's going to start to add RNA bases, okay? And when it makes RNA bases, the first thing that it does is that it starts to add nucleotides to make that mRNA. So RNA polymerase adds nucleotides to make the mRNA. It's kind of like DNA replication where A pairs with T, G pairs with C, but there is an exception. There is no such thing as T anymore. So instead of a T, it's a uracil, a U. So A pairs with U, C pairs with G, okay? If you ever see a T, it's going to be an A. You just remember that T does not exist in RNA. Okay, so once you're done making the copy, the DNA returns to its double helix form, and the mRNA is ready to leave the nucleus. Okay, once it's been edited, once it's been processed, it leaves the nucleus. Okay, and here's a question. What do you think is going to happen next? And what will the mRNA do now that it's been created? So here's your DNA. Just review DNA. You're making RNA here, so it's in the middle. And here's your single-stranded mRNA strand. It's now allowed to leave the nucleus, and it goes out to the cytoplasm. What happens next? So what is going to happen is the message is going to be decoded, and you are going to start forming a chain of amino acids. This process is called translation. You're translating the code into something that makes sense, which are your amino acids. So it says here, translation is to translate something. It means to write. To translate something means to write something in a different language. Okay, for example, English to Spanish, you're translating it in a different way. So why do we need to translate? And so again, you have your mRNA, and the mRNA has these bases, and what we need to do is convert them into amino acids. So for every 3 bases, you make 1 amino acid. So proline is an amino acid, methionine is an amino acid, serine is an amino acid, valine, leucine, these are amino acids. So again, to repeat myself, RNA is made up of nucleotides, A, U, G, C, and then these nucleotides, for every 3, you're going to make 1 amino acid. And then when you have a bunch of these amino acids, then we call that molecule a protein. So this right here is a protein. These right here are your amino acids. So translation, what happens is that the DNA was converted to RNA in transcription, and now in translation, mRNA is going to leave the nucleus and it's going to bind to this ribosome. Okay, so this is our ribosome, this is our mRNA. So we zoomed in, and for every 3 bases, you're going to make 1 amino acid. So you have to look for the start. The start signal is AUG. So the ribosome is gonna read. It's gonna go through and read each triplet until it finds AUG. So here it found AUG. This is the start. So it's gonna start the process of translation. Here comes a protein. Not a protein. Here comes a molecule called tRNA. tRNA is gonna come in, and it's gonna bring the appropriate amino acid to the ribosome. So here, the tRNA, which carries a leucine, it's like a taxi, it carries its passenger. It's gonna bring it in, and then it's gonna leave, and then another one comes in with another passenger, and then it's gonna leave. Then another taxi comes in, the ribosome reads the triplet, and then you form another amino acid. So you start forming this chain, and eventually the ribosome is going to read a stop signal. There are 3 stop signals: UAA, UAG, and UGA. Anytime it reads through one of those, it's gonna stop, and the chain is gonna cut off. It's gonna be cut, and that is called translation. We're basically making a protein. So these amino acids are then folded, and when they're folded, it forms a protein. So here's just a little bit of review. The mRNA comes in, the ribosome comes in, the tRNA is like a taxi. It carries the appropriate amino acid, and you start to form a chain. So a newly synthesized amino acid chain, and that chain is your protein. Okay, so I would recommend rewatching the first 9 minutes of this podcast, just because it's a lot and it's a little confusing. So if you listen to it one more time, it should click. Now, do you guys remember when I said that every 3 bases codes for 1 amino acid? The way we do that is we read something called a genetic code, and you always start where it says 1, and then 2, and then 3. And when you follow those 3 areas, you will land on a specific amino acid. So we're gonna try that right now. So here's another version of the genetic code. If we look at an mRNA, it says AUG. That's the first 3. So I'm gonna go here where it says first. This is A right here, and then U is right here. So you're going to follow where both of those connect. So U and A connect here in this box right here, and then you look at the third one, G, and you're gonna look for G, but you're going to go back to that box. And I noticed that the amino acid is methionine, MET. So MET, that's your first amino acid. It's gonna form a chain. And then we look at the next three, CCA. So C, and then here's the box where they lands. And then you look for the A. So on the third one, A is right there, and it lands under proline. So PRO is the next one. And then the last three is UAA. So UA, so this box, and then A, it will land under stop. So here it stops. You have two amino acids that formed, 1, 2, and when you fold it, you form a protein. Okay, so I'm gonna do the next one again. You could try it on your own, pause it, see if you could do it. Okay, now that you tried, you start with A, and then you go U, and then G. So that's gonna be methionine right there. Then the next three, G, G, C. So G, and then they both connect here on this box. And then you look at the row, G, so glycine. And then the last three, UGA, U, G, A, and it's gonna land on stop. Okay, now you're gonna do the last one on your own. See, I'm gonna give you the answer because I want you to see if you are getting it right. Okay, so MET, SER, and then stop. See if you get that for the third one, and if you did, then you're pretty much set. Okay. With the study guide, go ahead and fill in the questions. You're gonna write questions, and you're gonna write a summary at the end. Make sure you submit that.